So today we're going to look at the blood as part of the cardiovascular system. Your cardiovascular system is going to be what transports things in the body. It's going to include the heart, which is the pump, the blood vessels are the conducting system, and then the blood is the fluid medium, which we're looking at today. The job of the cardiovascular system is to transport things to and from the cells, things like oxygen, carbon dioxide, nutrients, hormones, the various immune system components, and then to be able to haul the waste products away. We have two different types of fluid that gets moved by this. The blood is going to be your plasma, and then the variety of cells that are in there that are going to transport nutrients and waste. We also have the interstitial fluid that's going to bathe the tissues. It will move nutrients and oxygen. It will diffuse from the bloodstream into the interstitial fluid and then into the cells. So it's going to move the waste in the reverse direction. It's going to help to haul it away. So hematology is the study of the blood and blood disorders. So the process of blood formation is hematopoiesis. So we have a few different terms for blood formation. Hematopoiesis is blood formation in general. Erythropoiesis is just specifically the red blood cells. And then another H word in here, hemorrhage, is the loss of blood. So blood is a fluid connective tissue. It's going to help to transport dissolved gases, nutrients, hormones, and waste products. It's part of the system that will help to regulate your pH and ion composition. It will help to restrict fluid loss at injury sites, and it plays an important role in the body's defenses against various toxins and pathogens. And then it will be helpful in regulating body temperature. So if you've ever experienced circulation being cut off in an area of your body, you'll notice it will cool off pretty quickly in there. So it will help to absorb and redistribute heat. So the physical characteristics of blood, it is more viscous than water and it flows more slowly than water. The temperature of the blood is 100.4 degrees Fahrenheit or 38 degrees Celsius. That is a little bit above what you see for normal body temperature. Normal body temperature, we're usually measuring on a mucous membrane. To measure the temperature of the blood, you would have to insert a thermometer into the body. And typically, that's not something that is very pleasant for a human. We don't really like to be poked with meat-type thermometers. The pH of the blood is about 7.4. It makes up about 8% of the total body weight. So typically, for a male, you're going to have 5 to 6 liters of blood. A female is 4 to 5 liters. This is dependent on your size, so that is an average. But there is a hormonal negative feedback system that wants to make sure you maintain a consistent blood volume and that the osmotic pressure stays consistent. So when we look at a sample of whole blood, it's going to have the plasma, which is going to be 40 to 63%. And then in the plasma, you've got plasma proteins, other solutes, and water. Water makes up 92% of that. And then your formed elements are 37 to 54%. When we look at those plasma proteins, albumin is going to be the majority of it that helps to contribute to your osmotic pressure. Then you'll have the globulins, which are immune proteins, fibrinogen, that's helpful with clots, and then other regulatory proteins in there. We do have some other solutes, your electrolytes, various other organic nutrients in there. So it can be fatty acids, cholesterol, carbohydrates like glucose, other amino acids, and then your organic wastes that are going to eventually get excreted, things like urea, uric acid, creatinine, bilirubin, ammonium, that will be found in the blood. So plasma, over 90% of it is water. 7% of it is plasma proteins. They're made in the liver, and they are confined to the bloodstream. So albumin is the one that's going to maintain the blood osmotic pressure. So albumin being off could be an indication of something affecting the liver potentially. It could be as simple as a person had IV fluids that would throw off the balance temporarily. Your globulins or immunoglobulins are going to be antibodies that are going to bind up things to form your antigen-antibody complexes. And then you have transport globulins, things like hormone-binding proteins, the metalloproteins, and that would bind metals, the allolipoproteins that would bind lipids, and then steroid binding proteins. So here in this little test tube, you can see 55% is plasma, 45% is the cells typically. And then this little white layer here they call the buffy coat is where you're going to have the white blood cells and platelets. So fibrinogen is used to produce fibrin that is used for clotting. So other plasma proteins are about 1%. They will vary in their quantities. You can have enzymes, hormones, and prohormones that are used to make hormones. Most of them are going to come from the liver. Antibodies will come from your plasma cells. Peptide hormones will come from your endocrine glands. And then the serum liquid part of a blood sample is going to have your dissolved fibrinogen that can later be converted into solid fibrin. Plasma is just going to be that liquid part minus the clotting proteins. So other substances make up about 2%. They would include electrolytes, nutrients, gases, and other waste products. Formed elements, these are your cells. The red blood cells, or erythrocytes, their job is to transport oxygen. Erythro means red, cyte is cell. So these are the red blood cells in this illustration here. Your white blood cells, or leukocytes, are the ones that are going to play an immune function. Leuko means white. The granular leukocytes are going to have granules in them, and so they have a more granular appearance. They're going to include the neutrophils, eosinophils, and basophils. Agranular do not have the granules in them. So your lymphocytes, they are produced in lymphoid tissue. They are your B and T cells, as well as the natural killer cells. We also have monocytes, platelets. Platelets are actually little fragments that we use for clotting. Only the white blood cells are complete cells. Red blood cells have no nuclei and they don't have any organelles. They're really just cell fragments, but they work to do their job. Their job is mainly to transport oxygen. Most of the formed elements will live in the bloodstream for only a few days. So most of the blood cells do not divide, but they are produced by stem cells in the bone marrow. So here's an electron microscope picture of some red blood cells and some platelets. And then up here we've got some illustrations. You can see the platelets, monocytes. Monocytes have this C-shaped nucleus in there. The neutrophil, also called a polymorphonucleocyte because it will look like it's got multiple nuclei to it. The eosinophils are kind of a reddish color. Basophils are blue. And then the B and T lymphocytes look a lot alike. So this picture here shows where these are derived from. You start with this pluripotent stem cell, and it's got two choices. It can become a lymphoid or a myeloid stem cell. If it becomes a lymphoid stem cell, those will lead to producing your B and T lymphocytes. The myeloid stem cells, those will lead to producing your red blood cells. It'll make megakaryocytes that eventually become platelets, eosinophils, basophils, neutrophils, and your monocytes, which can become your macrophages. So we have little hormone signals that are going to help to tell them which pathway they should go on in their development. So we have too many red blood cells to have it be practical to actually count them, and you'd have to remove them from the patient, which isn't good either. So instead what we do is a hematocrit, which is going to look at the percentage of the blood that's actually red blood cells. So in a male, we have between 40 and 54%. In a female, we have 38 to 46%. So why look at this? It can give us an indication of the status of red blood cell production, how hydrated we are. Abnormally high hematocrits can be seen when a person's been in a place of higher altitude. So with hypoxia, it can stimulate that. With athletes, you could see this being high if they've been doing blood doping, where they would save and bank some of their cells and put them back into their bloodstream. It can be altered with polycythemias, anemias, hemorrhage, malaria, cancer, chemotherapy, and radiation, and various drugs can affect that. So your erythrocytes, these are the most numerous of the formed elements. For a female, you're going to have between 4.3 and 5.2 million cells per cubic milliliter. For males, 5.2 to 5.8 million cells. They have a unique shape. It's a biconcave disc. And this folding allows it to have more surface area. The plasma membrane contains a protein spectrum that helps to give it more flexibility. So you're able to have a cell that's 7.8 micrometers pass through a 4 micrometer capillary because they can fold. So anucleate, there's no centrioles, no organelles, so they don't do any cell division. No mitochondria means they will generate ATP anaerobically. This is actually a good thing. It prevents it from consuming the oxygen that it's transporting. So if it had an interest in that oxygen, it wouldn't make it as good of a transport tool because it would want to use the oxygen instead of transporting it. Being that it doesn't use it, it's just content to transport that oxygen for you. It's filled with hemoglobin, so that's 97% of the cell's contents, and this is going to help to function in gas transport. So what happens is you have hemoglobin and oxygen that are separate. They come together to form oxyhemoglobin, and then it will separate again when it wants to release it in the tissues. So the formation of blood cells, or hematopoiesis, is important. The leukopoiesis is specifically to make leukocytes, erythropoiesis specifically to make red blood cells, and then thrombopoiesis is specifically for making the thrombocytes. This does occur in the red marrow, which is in the spongy spaces of your compact bone, or in the spongy spaces on the ends of your bone. So it's going to have compact bone around it, but you will have the spongy bone in the middle with the red marrow. So in adults, places where this would happen would be the proximal epiphysis of the femurs, and then the bodies of the lumbar vertebrae are good locations for this to happen. So they originally start out as that pluripotent stem cell that's made from mesenchyme tissue. So in the fetus, hematopoiesis is going to occur in the liver, spleen, thymus, lymph nodes, and yolk sac. So a fetus does not have the developed bone with the red bone marrow to be able to make it in the marrow. Erythropoietin is a hormone that's going to come from the kidneys, and that's going to stimulate hematopoiesis. So the things we need to be able to make erythrocytes, we need to have the appropriate amino acids, iron, vitamin B12, B6, folic acid, and then an adequate supply of carbohydrates and lipids. So this little chart here shows the development that erythrocytes go through. They start out as a hemocytoblast, become a proerythroblast, the early and late erythroblasts, the normoblast, reticulocyte, and finally the erythrocyte. So erythrocytes, their job is to transport the respiratory gases. Hemoglobin is what allows this to happen. It's going to reversibly bind with oxygen. Most of the oxygen in the blood is going to get bound up to hemoglobin. Hemoglobin is made up of the protein globin, and then it's got 2 alpha and 2 beta chains. So with the heme molecule, it's going to bear an atom of iron, which can bind to 1 oxygen molecule. So each hemoglobin molecule is able to transport 4 oxygen molecules as oxyhemoglobin. So each red blood cell is going to have, on average, 250 million hemoglobin molecules. Each hemoglobin molecule is going to have 4 oxygen-binding sites. So a single red blood cell can transport about 1 billion oxygen molecules. Carbon monoxide, however, is a problem for us. It's going to have about 200 times the affinity for hemoglobin that oxygen does. So what it will do is bind to the hemoglobin and not let go and take up the space that could be used for oxygen transport. So it can actually fool oxygen saturation detectors. Fetal hemoglobin has to be a little bit different. It's a strong form of hemoglobin, specifically in the embryo. It needs to be a little bit stronger so that it can take oxygen from the mother's hemoglobin. It just needs to have a slight advantage there. Carbaminohemoglobin with low oxygen, this will have the hemoglobin release the oxygen, and then it will bind the carbon dioxide in the tissue and carry it to the lungs. So this is an important step, because once we have hauled the oxygen to the tissue, we need to drop it off there. This allows that to happen and then to pick up the waste product of the carbon dioxide. So anemia occurs when your hematocrit or hemoglobin levels are below normal. There are several different types of anemia, and it can be caused by several different conditions. So red blood cells, we make a lot of them and go through a lot of them, so we do recycle them. So a typical red blood cell will live about 120 days. So what will happen is when you have low oxygen levels, your erythropoietin would stimulate the production of red blood cells. That's going to happen in the marrow. And then your old red blood cells are going to be broken down. They will be engulfed by the macrophages in the liver, the spleen, and the bone marrow. And you're going to keep the hemoglobin and break it down. So with the hemoglobin, you're going to have the heme portion that will end up making bilirubin. That will be used as a pigment for stercobilin, for pigmenting the feces, urobilin to pigment the urine. And then it will also make bile that is stored in the gallbladder, and that is used for digesting fats. The iron is going to get bound up as ferritin and hemosiderin. You want to bind up that iron so that it's not available to microbes. And then the globin portion will just be broken down into the individual amino acids and be able to be reused in the body as well. So this is another picture that demonstrates that happening in there. Use whichever one makes more sense to you. So typically that red blood cell is going to live 100 to 120 days. It will travel about 750 miles in that time, which would be like going from Los Angeles to Albuquerque, New Mexico. So we do need to recycle the old erythrocytes because they become frigid and fragile. Their hemoglobin starts to degenerate. They're just not going to do a good job anymore. So they will be engulfed by the macrophages. So again, heme and globin are separated. The iron is removed to be reused. So for females of childbearing age, they do need to have slightly higher iron intakes because the red blood cells from the menstrual cycle are not recycled. So we can have the transferrin that will transport it. It can be stored as hemosiderin or ferritin in the tissues. The heme will make your bilirubin, can be used for the bile, urobilin, and then the stercobilin in the feces. So erythropoietin, that will be produced when the peripheral tissues are low in oxygen or hypoxic. One way to stimulate this to happen is to go up in high altitude. It takes about 2 weeks for a person to adapt when they go up to altitude. There are diseases that can cause that to happen as well. So hemoglobinuria is when you have the hemoglobin products in the urine. So that can be due to excess hemolysis in the bloodstream. So hematuria is when you have whole red blood cells in the urine. This would be due to kidney damage or tissue damage. And then with anemia, it's having insufficient erythrocytes. One type is hemorrhagic. That's due to a loss of blood from something like bleeding. Hemolytic anemia, here the red blood cells are ruptured too early. Aplastic anemia, they're being destroyed or inhibited in the red bone marrow. So when you have anemia, you're going to decrease the hemoglobin content. So with iron deficiency anemia, that's a secondary result of something like hemorrhagic anemia or inadequate intake of iron in the diet or having impaired iron absorption. So the trick is to figure out what is the cause of it. With pernicious anemia, this is a deficiency of vitamin B12. So this can be from a lack of intrinsic factor that's needed for the absorption of B12. So in those cases, they give intramuscular injections of vitamin B12. So then you can also have abnormal hemoglobin. So the Thalassemias, these are going to have an absent or faulty globin chain in the hemoglobin. This is going to result in red blood cells being thin, delicate, and deficient in hemoglobin. With sickle cell anemia, this is going to be the result of a defective gene that codes for normal hemoglobin. So it has a single amino acid substitution in the beta change, and it causes the red blood cell to become sickle-shaped, hence the name. And that will happen in low oxygen situations. So hypoxia is when there is insufficient oxygen. With polycythemia, this is having excess of red blood cells. And that's not a good thing either because that's going to increase the blood viscosity. You want to have just the right amount. So the three main types of polycythemias are polycythemia vera, secondary polycythemia, and then the result of blood doping. So polycythemia vera, you're making too many of them. Secondary, there's something else that's caused it. Blood doping, you put them in there. So we do have homeostatic mechanisms that do try to maintain the level of red blood cells in there. So if you have some sort of disruption in there that can impair this, it can lead to changes in oxygen delivery to the kidneys. The kidneys detect that low oxygen, and they're going to secrete the erythropoietin into the bloodstream. That's going to cause the red marrow to produce more red blood cells. You get more red blood cells in the circulation, and then you'll have more oxygen going to the tissues. So it will provide a negative feedback loop and shut things down when oxygen delivery is adequate. So here are some typical tests performed for red blood cells. So we already talked about the hematocrit that's going to look at what percentage are red blood cells in there. You can look at the reticulocyte count, the hemoglobin concentration, total count of red blood cells, the mean corpuscular volume, and the mean corpuscular hemoglobin concentrate. And then you can see things that go with these. For almost all of them, if they're low, it's a type of anemia. So for human blood groups, the red blood cells are going to have these glycoprotein antigens on their surface. These antigens are going to be unique to individuals. So if you put them in somebody else, they're recognized as foreign. So the agglutinogens are going to be promoters of agglutination. So the presence or absence of these antigens is what we use to do blood group classifications. So we hear about the ABO blood typing and the RH blood typing all the time. But in humans, we actually have 30 varieties of naturally occurring red blood cell antigens. The ABO and RH groups are the ones that are going to cause vigorous transfusion reactions when they're improperly transfused. So some of the other groups, we have the M, N, Duffy, Kell, and Lewis antigens. And those are a lot of times used just for identification purposes for legal things. So with the ABO blood groups, you've got an A antigen and a B antigen that can be on the surface of the red blood cells. We also can produce antibodies to these. So you can have an anti-A antibody and an anti-B antibody. So you can have different patterns of these. So if you are type A, you're going to make surface antigen A. You're going to make antibodies towards B because you don't produce B. If you are type B, you're going to have the B surface antigen, and you're going to make antibodies towards A because you don't have that. If you are AB, you're going to have both the A and the B antigen on the surface, and you won't make antibodies to either one of them because you have both of them. If you are type O, you don't have any of the markers on there, but you'll make antibodies towards both the A and the B. So if you're looking for the type of blood that would have the most universal application for donation, type O, because it doesn't have any antigens on there. If you're looking for the person who can take the most types of bloods, that would be an AB person because they're not going to produce the antibodies. So you could put a type O blood in a person with type A because even though they have the anti-B antibodies, there is no B antigen on there. So this is just showing what would happen is if you put in red blood cells that had an opposing antibody, you would have agglutination and then hemolysis. So this gives approximate frequencies of the different blood cell types in the US population of different ethnicities in here. So with type O is fairly common in a lot of people. Type A, B is less common, and then A and B are somewhere in the middle. So another marker we have on the blood is the Rh group, sometimes called the D antigen. Rh comes from it being found in rhesus monkeys. About 85% of the population is going to have this, so we say that they are Rh positive. If they lack the antigen, we call them Rh negative. That would be about 15% of the population. So the anti-Rh antibodies are not spontaneously formed. But what happens is if an Rh negative person gets blood from an Rh positive person, the anti-Rh antibodies are going to form then. First time, probably not going to be that much of an issue. But the second exposure, that's when you're going to get the bad transfusion reaction because the cells have been sensitized. So only sensitized Rh negative cells, when you put in the Rh positive blood, are going to have an aggressive reaction with the Rh antibodies. Where this is most commonly an issue is when we look at hemolytic disease of a newborn infant. If you have an Rh negative mom who is going to have a baby with an Rh positive man, the baby is going to be Rh positive because it's the dominant trait. The mom will form anti-Rh antibodies. So during subsequent pregnancies, even if it's a different Rh positive man, the anti-Rh positive antibodies are sensitized, and they'll be able to cross the placenta and destroy the red blood cells of that Rh positive baby, which is bad news for the baby. So what they do is give the drug RhoGAM that will prevent the Rh negative mother from becoming sensitized. So if this does happen, they try to treat the hemolytic disease of the newborn. It would sometimes take pre-birth transfusions and exchange transfusions. It's really not a position you want to have that baby in. So the RhoGAM injection is very helpful there. So this is just showing here how you can have the fetal tissue and the maternal tissue allow the blood cells to be able to cross and interact with each other. So transfusion reactions occur when mismatched blood is infused. There is always some risk with a blood transfusion, but they do test matches before putting the blood in for a person. So the donor cells are attacked when the recipient's plasma agglutinins would attack. It can cause diminished oxygen carrying capacity. It can clump the cells, and that's going to impede blood flow. And then it can rupture red blood cells, and that will release free hemoglobin into the bloodstream, which is going to be a problem. So that circulating hemoglobin can precipitate in the kidneys and lead to kidney failure. So that's why they do the blood type or the cross-match test. So your leukocytes are white blood cells. They don't have hemoglobin, but they are going to have nuclei and the other organelles, so they're a complete cell. What white blood cells do is they're important for immune responses. They're going to defend against pathogens, help to remove toxins and waste, attack abnormal cells. Most of them are going to be in the connective tissue proper in the lymphatic systems and organs. We don't have nearly as many of these, so 6,000 to 9,000 per microliter. They migrate out of the bloodstream via diapedesis, and then they can move through the tissue using amoeboid-type movements. A lot of times they are going to be attracted to chemical signals, so we call it positive chemotaxis. Some of them are phagocytic, so these would include the neutrophils, eosinophils, and monocytes. So leukocytosis is going to occur when you have a white blood cell count that's over 11,000 per cubic millimeter. So this is a normal response to a bacterial or viral invasion. So it's not always a bad thing. If you have an infection going on, you want to have this type of response. Leukopenia is when you have a white blood cell count that's below 4,800. And then leukemia is when you have cancer of the white blood cells. So here we can see some pictures of what some of these look like. So the neutrophil, you can notice the multiple lobes. Eosinophil, you'll notice the reddish color in the dye. So the basophil, some of them appear more purplish, some appear more blue. Here you have the monocyte. This has this big C-shaped nucleus in there. You've got a platelet down here, the little fragment, and then you've got a lymphocyte here. So granulocytes, the granular appearance because of those granules, they end in phils. Neutrophils, eosinophils, and basophils. Agranulocytes will be your monocytes and your lymphocytes. So here you can see a nice eosinophil, a nice PMN. So white blood cells roll along the endothelium, which is going to be the lining in the blood vessels. They can stick to it and then squeeze between the cells. So these adhesion molecules, the selectins, help the white blood cells stick to the endothelium. This will happen near sites of injury. The integrins on the neutrophils help move them through the walls. So your neutrophils and macrophages are going to be good for phagocytizing bacteria and debris. So when they get a chemical signal that would attract them, we refer to this as chemotaxis. So this can happen from both the kinins and injury sites, and toxins can do this. So diapedesis is the passage of the blood cells through the intact capillaries. So it is a normal process to go through an intact capillary wall, but it will usually occur when there is inflammation in there. So then they're able to squeeze through there. So this is showing one squeezing through the cell walls. So granulocytes, they have cytoplasmic granules that they are named for, and they stain specifically different colors with right stain. So they're larger, they're usually short-lived, they don't live near as long as a red blood cell, they'll have lobed nuclei, and all of them are phagocytic. So the most common of these is the neutrophil, sometimes called a PMN, a polymorphonucleoside. It's sometimes also called a SEG. So they're 50 to 70% of those circulating white blood cells. The nuclei will look like it's got 2 to 5 lobes. Older cells will have more lobes. The young cells are sometimes called band cells because they will have a horseshoe-shaped nucleus. So they have pale cytoplasm granules. It's kind of a pale lilac shade. In there, you'll have lysosomal enzymes, bactericides like hydrogen peroxide and superoxide. These are going to be the ones that are going to respond fast. They're the first to attack bacteria. They do elevate fairly easily. So it's one of the things can be checked for to see is there a possible infection, but they can also elevate from other things like trauma to the area. So they'll engulf pathogens, digest them, they release prostaglandins and leukotrienes, help to form pus. So degranulation is when you remove those granules from the cytoplasm. So defensins are peptides that are going to come from the lysosomes, and they help to attack pathogen membranes. The eosinophils are only 2 to 4% of the circulating white blood cells. They can attack large parasites. They'll excrete toxic compounds, things like nitric oxide and cytotoxic enzymes. A lot of times the parasite is just too big to engulf entirely. So they do have red-stained bilobed nuclei, and they will have a broad band of nuclear material, but they can have up to like 3 lobes. So they'll stain red to crimson because they're acidophilic. They're large, coarse, lysosome-like granules, but the granules are not going to actually obscure the view of the nucleus. You'll still be able to see that. So these are important for parasitic worm infections to help to attack them. They are involved with allergic reactions. So what they can do is help to phagocytize antigen-antibody complexes. And then they can leave the capillaries and enter the tissues and release histamine. So part of what is going on with an allergic reaction is they can be releasing some histamine in there, which if you have allergies, you know it's pretty unpleasant stuff to have to deal with. And then they can slow down the inflammation caused by basophils. Basophils are less than 1% of the circulating white blood cells. They will have a U or S shaped bilobe nuclei with 2 or 3 conspicuous constrictions in there. So they are large with a purplish black granules in there that can obscure the nucleus, and they can accumulate in tissue damage. So things basophils are going to be involved in is inflammatory and allergic reactions. They will leave the capillaries and go into the connective tissue as mast cells. In there, they can release heparin, histamine, and serotonin that help to increase that inflammatory response and give you your hypersensitivity or allergic reaction. So histamine is an inflammatory chemical. It acts as a vasodilator, so it's going to allow more blood flow into the area and more fluid. Other white blood cells can come in. That is why an antihistamine is taken in these situations. This is only one of the chemicals that is involved with this response. So if you have allergies and you've taken antihistamines and you're like, wait a minute, I take that antihistamine and it helps, but it doesn't eliminate the allergy, it's because you're only attacking one aspect of it with the antihistamine. There still is other things promoting allergic responses. So heparin is another hormone that will help to prevent blood clotting. So those agranulocytes, they lack visible cytoplasmic granules. Similarly structured, but they do have functionally distinct and unrelated cell types. So they can be spherical like the lymphocytes or more kidney-shaped like the monocytes nuclei. So monocytes are 2 to 8% of the circulating white blood cells. They are the largest. They stain purple with the U shape or kind of a C shape depending on which angle you're looking at it from, nuclei. They're able to leave the circulation and go into the tissue, and then they would differentiate to become a macrophage. So macrophage literally means big eater. They do take a bit longer to get to the site of infection, but they come in large numbers, so they bring friends. They become wandering macrophages once they leave the capillaries. So they're able to destroy microbes. And then they can help clean up the dead tissue following an infection. They'll engulf large particles, various pathogens, and then they can secrete substances that are going to help bring in other cells of the immune system and fibroblasts to areas of injuries. So your lymphocytes, they're 20 to 30% of the circulating white blood cells. They are a little bit bigger than the red blood cells, and they'll go in and out of the blood. They're found mostly in the connective tissue and the lymphatic organs. They like to hang out in the lymphatic tissue to be able to wait for the pathogens to come through. So these are part of the body's specific defense system. So you have general immune defenses that are geared towards anything to try and just give an overall layer of protection. Specific defenses are very specialized towards a particular invader. So these will generally increase during viral infection. There's 3 classes of them. So the first class is the T cells. These will give your cell-mediated immunity. They're going to attack the foreign cells directly. A lot of times those foreign cells want to get inside our cells, so they're attacking that. B cells are responsible for more of your humoral immunity. They'll become a plasma cell that's able to synthesize antibodies. And then the natural killer cells, or NK cells, they detect and destroy abnormal tissue. So these are really useful for attacking cancer cells. They are your friend. So when doing some blood tests, if you're just looking at red blood cells, you don't necessarily want to pull up everything with the white blood cells. But if you do, you usually get it with a differential count. That will detect the changes in specific white blood cell populations. So you can see which type of white blood cell is elevated. This can be useful in diagnosing infections, inflammation, or allergic reactions. So we do have some white blood cell disorders. Leukopenia, that's an abnormally low white blood cell count. Leukocytosis is when it's high. With leukemia, it's going to be a high white blood cell count. It's a blood cancer, white blood cell cancer. So the differential is able to help you detect the changes in these numbers and help you determine, is there an infection, poisoning, leukemia, chemotherapy going on, parasite infection, or allergic reactions. So our numbers here, neutrophils typically 60 to 70%. If it's a bacterial infection, they're going to go up. Lymphocytes, 20 to 25%. They would go up with a viral infection. Monocytes, 3 to 8%. Those would go up with viral and fungal infections. Eosinophils, those are elevated when there is a parasitic response or an allergic response. And then basophils, they are less than 1%. They can go up with allergic reactions, or they may be elevated in a person with hypothyroid. So again, when we look at the blood cell formation here, you're going to have your myeloid or lymphoid stem cells. The lymphoid stem cells make your lymphoblasts. This is going to make your lymphocytes. Everything else is going to come from a myeloid stem cell. So your erythrocytes, the platelets, basophils, eosinophils, neutrophils, or monocytes. So in here, these are the little chemical signals that are going to determine which one do you need to make. So when you've got this myeloid stem cell and it's waiting to find out what it's going to be when it grows up, it's waiting for some of these chemical signals to come along and say, okay, nope, EPOs come around, you need to become a red blood cell. So all of those blood cells originate from the hemocytoblast. So the myeloid stem cells are going to produce everything except the lymphocytes. Lymphocytes come from lymphoid stem cells. Lymphopoiesis is the production of the lymphocytes. So all of them, except the monocytes, develop in the bone marrow. Monocytes will develop in the peripheral tissues into macrophages. So some of the lymphoid stem cells can migrate to peripheral lymphoid tissues like the thymus, spleen, or lymph nodes. So colony stimulating factor is one of the hormones that regulates the blood cell populations. So MCSF is going to stimulate monocyte production. So GCSF is going to stimulate granulocyte production. GM-CSF will stimulate granulite and monocyte production. And then multi-CSF will accelerate the production of the granulocytes, monocytes, platelets, and the red blood cells. So again, some more different pictures here in this diagram that you're welcome to take a look at. With any of these diagrams, I did leave the notes as Word documents so that you can enlarge them if you would like. Platelets, these are cell fragments that we use for clotting blood. So we see these in the humans. Non-mammalian vertebrates have thrombocytes, which are nucleated cells. So they're made from flattened discs. The platelets will circulate for 9 to 12 days, and then they'll be removed by phagocytes in the spleen. We keep about 2/3 of them reserved for emergencies. So thrombocytopoiesis is platelet production. This would occur in the bone marrow. You would have the myeloid stem cells make a megakaryocyte colony forming cell that becomes your megakaryoblast and then your megakaryocyte that's going to break apart, and that will create the cell fragments that become the platelets. This is stimulated by thrombopoietin, thrombocyte stimulating factor, IL-6, and multi-CSF. So interleukin, we just abbreviated as IL-6. It's a cell or a chemical used for blood cells to communicate. So platelets, they will release important clotting factors. They temporarily patch up a damaged vessel, and they can cause the tissue to contract after clot formation. So typically you'll have 150 to 500,000 platelets per microliter. That's normal. So with a complete blood count, this can be used to screen for types of anemia and infection. The differential will tell you the specific proportions of the white blood cells. And then when you look at the hematocrit and hemoglobin measurements, it's going to tell you indications about the red blood cell health. Normal hemoglobin ranges in an infant is 14 to 20 per 100 milliliters. An adult female will have 12 to 16 grams per 100 milliliters, and an adult male 13.5 to 18 grams per 100 milliliters. So hemostasis is that stop of blood loss. It stops the bleeding. It's quick, it's localized, and it's to prevent hemorrhage. Hemorrhage would be a large amount of blood loss. There's 3 phases: the vascular phase, platelet phase, and coagulation phase. So during the vascular phase, you have damage to the blood vessels. This will stimulate pain receptors. There's a reflex contraction of the smooth muscle to the blood vessels. What it's trying to do is reduce the blood loss out that area, and that will last from 30 minutes to several hours until other mechanisms can take over. That works well for small blood vessels or arterioles. So when the endothelial cells contract, it's going to expose the basal lamina to the bloodstream. And those endothelial cells will release chemical factors like ADP, tissue factor, and prolactin. You'll also have local hormones like the endothelins that will stimulate the smooth muscle contraction and cell division. So those endothelial cell membranes become sticky, and that allows them to seal off the blood flow in there. So when you've got the injury, you create the vascular spasm in here. So you can see you can have the concentric smooth muscle flow. Here's your vessel wall, the basal lamina, and the endothelium. So the platelets, you're going to have platelet adhesion where it's going to start to stick to the tissue around it. The platelets will aggregate to form this platelet plug. And here's your cut edge of the vessel wall where the plug gets in there to block things up. So the platelet phase happens within 15 seconds of injury. So platelets themselves store a lot of granules needed for the platelet plug formation. We call them the alpha granules. There's clotting factors in there, platelet-derived growth factor that will cause a proliferation of the vascular endothelial cells and smooth muscle, and the fibroblasts to get damage repair started. And then you've got dense granules in there. You also have ATP, ADP, calcium, serotonin, fibrin stabilizing factor, and enzymes that are going to help with the production of thromboxane A2. So we have your 3 steps: the platelet adhesion, where they're going to become sticky, the release reaction to call more of them into the area, and then they will aggregate and form your clump or plug. So during platelet adhesion, platelets adhere to the sticky endothelial surfaces, the basal lamina, to expose collagen fibers underlying damaged endothelial cells in the vessel wall. So here are some damaged collagen fibers in there that those platelets are going to want to stick to. The platelet release reaction you're going to release thromboxane A2 and ADP to activate other platelets. The serotonin and thromboxane are vasoconstrictors that help decrease blood flow through that injured vessel. So these become activated when you have the adhesion extended projections make contact with each other. So with platelet aggregation, those platelets stick together, helps to activate new platelets, and makes your plug to close off small breaks. And then this is reinforced by fibrin threads during the clotting process. So you've got your plug, and then you have these fibrin threads that are going to form in there to help reinforce that. So prostacyclin is going to come from those endothelial cells. It will inhibit platelet aggregation. You have other inhibitory compounds that are released by white blood cells. You have circulating enzymes that will break down the ADP, and then negative feedback from serotonin. So you can develop the clot to help to isolate the area. So the coagulation phase, this begins about 30 seconds after injury. So the blood drawn from the body starts to thicken into a gel, and this will separate into a liquid and a clot of insoluble fibers from the fibrin that gets trapped. So if clotting occurs in an unbroken vessel, we call it thrombosis. So some chemicals we need for clots would include calcium, enzymes from the liver cells, and then other substrates from the platelets or damaged tissue. It's actually a clotting cascade of reactions where each clotting factor is going to activate the next step in a fixed sequence that will end up triggering the formation of these fibrin threads. So prothrombinase and calcium help to convert the prothrombin into thrombin, and that turns the fibrinogen into fibrin. So the clots will end up creating a fibrin network over the platelet plug that helps to trap those cells in there and seal off the area. So we have this common pathway that is shared. You have an intrinsic and extrinsic reaction. So extrinsic here, you have some tissue damage. You're going to have tissue factor 3. Calcium comes in, clotting factor 7. Factor 7 with the tissue factor complex comes in, to factor 10, which is the start of the common pathway. For the intrinsic pathway, you have activated proenzymes, so it's usually factor 12, platelet factor 3, calcium comes in, clotting factors 8 and 9, and then factor 10, activator complex. So once you get to this factor X, then you have prothrombinase, prothrombin, thrombin, the fibrinogen comes in, and you make the fibrin. So extrinsic if it's from the outside, intrinsic from the inside, they come together in that final common pathway. So this shows a comparison again where you have the extrinsic pathway from tissue trauma, intrinsic pathway from internal blood trauma, so an endothelial cell exposed to the collagen fibers. So both of them do require calcium, and you end up getting to this activated factor 10, and you'll have prothrombinase. You come into the final common pathway to make prothrombin, thrombin, your fibrinogen, and your stabilized fibrin threads around your clot. So that extrinsic pathway, it's a vessel wall outside the bloodstream. The damaged tissue leaks the tissue factor, the thromboplastin, into the bloodstream. Prothrombinase is formed in seconds. You do need the calcium and the clotting factor 10 to combine with 5 to form prothrombinase. The intrinsic pathway, you're going to have coagulating enzymes within the bloodstream. So in the endothelium, they come in contact with the collagen. Your damaged platelets release the phospholipids. It does require several minutes for this reaction to occur. Substances involved, calcium and clotting factors 12, 10, and 5. So the final common pathway, the prothrombinase and calcium catalyze the conversion of prothrombin to thrombin. That's going to convert the soluble fibrinogen into insoluble fibrin threads. It activates the fibrin stabilizing factor 13 and your positive feedback effects of thrombin. So this will accelerate forming prothrombinase and activates the platelets to release phospholipids. So this is a nice electron microscope picture of a clot being formed. So you can see that with all these fibers in there, this is why scabs are kind of a rough-looking tissue. So the plasma clotting factors of procoagulants are important. You need these proteins and ions in the plasma in order for normal clotting. If you don't have them, then there is the risk of hemorrhage or bleeding. So both calcium and vitamin K are essential. So this just lets you see where these different things come from. So when you're done with the clot retraction and repair of the blood vessel. So the clot plugs the ruptured area for a while, but the platelets pull on the fibrin threads and it causes the clot to retract. And the trapped platelets release factor 13 that helps to stabilize those fibrin threads. It's going to pull the edges of the vessel together. So it's like it goes in there, makes the clot, and then it kind of shrinks up and pulls the tissue together to help repair the vessels. So clot retraction occurs after the clot's formed, and it takes 30 to 60 minutes. So eventually you need to get rid of the clot. Fibrinolysis is that process. And you're going to have the thrombin and tissue plasminogen activator activate plasminogen that will produce plasmid to digest the fibrin strands. So some pathologies that go along with blood clotting. Hemophilia, it's an impaired ability to clot. Most of the time it's hereditary, where the person is lacking a coagulation factor. The most common one that is impaired is 8. The thrombus is a clot formed in situ within the vascular system, and that's going to impede blood flow. An embolism obstructs the artery, is typically obstructed by a clot of blood or an air bubble. So thrombosis occurs when the thrombus or blood clot develops in the blood vessel and reduces blood flow through that vessel. The embolism is when the piece of the clot or a foreign object or some other substance becomes stuck in the blood vessel and obstructs blood flow. They're dangerous because they can lead to heart attacks, strokes. The tissue does not get proper blood flow for oxygenation, and that can damage or kill the tissue quite quickly. So that is the section on blood.